So welcome again. Thank you very much. So today we are here to discuss about a very interesting concept of DCEA. And DCEA stands for Dynamically Controlled Environment Agriculture. So if you don't know what is that, it's basically something like the urban agri or CA, but in a way that we can, in a lifetime there and then, we can actually change little things. If you can mute yourself would be great. If you have to ask something, just let us know. So DCA is basically a concept where in CA, in controlled environment agriculture, by recording and monitoring very closely different parameters and how plants behave, then you can adjust in a lifetime as you grow the environmental conditions and the inputs, and then to adjust basically your outputs based on what you want to achieve. And today we have with us Abby, who is the author of this recent paper talking about the concept of DCA. And we have also with us Professor Tony Pridmore, who is also working with plant phenomics and director of the network of Phenom UK. So the first part of this webinar, Abby and Tony will talk a bit about what they're doing and what this exciting DCA concept is all about. And then we'll open questions from the audience and some questions to Abby and Tony. So we'll start with Abby. Abby, over to you.
Thank you. So let me share my screen. And yeah, as Katia mentioned, we're here to talk about dynamically controlled environment agriculture. And this is we were featured on the cover of ES&T Engineering in November for our paper discussing the concept, integrating machine learning and mechanistic modeling for sustainable food cultivation. And the idea is that in conventional, or when we think about controlled environment agriculture, as opposed to what we propose in the paper about dynamically controlled environment agriculture, we generally think of a tightly controlled environment where we're growing produce, we're controlling the inputs, the environmental controls, parameters in the environment to lead to the best possible yield that we can achieve with those inputs. And the concept of dynamically controlled environment agriculture is that we use sensing and biofeedback, so that's kind of listening to the plant, watching the plant, to take measurements constantly. So we're still controlling the input parameters like light, temperature, nutrient concentration in the water. But at the same time, we're observing the plant's state at a given moment. So things like pigment, size of the plant, and the nutritious contents, nutrient density within the plant. And this is largely theoretical at this point in the state of plant science, but the idea is that with these, with a dynamic sensing environment, we can look at things like the pigments in the plants and determine what is the plant lacking, or what can we tune with the control inputs to get to a more desired outcome. And the desired outcome is generally in controlled environment agriculture thought of as the best possible yield, the most nutrient dense, or the best tasting. Too nutrient dense can get a little too flavorful for some people, so we like to balance the nutritious contents with the flavor, obviously. But the idea is that we can use things like visual sensing to take stock at a given time of what's going on in the plant, so that we can then change and control our environmental controls to yield a better outcome. And the reason that in the paper, we discuss dynamically controlling this environment instead of just having set points where, you know, this is the optimal concentration of nutrients for a plant versus taking stock as we go, is because there are a lot of different desired outcomes that we can talk about. So as I mentioned, we don't just care about the nutritious quality of the plant or how big it is. We also want to take stock of things like the material use and the water conservation and life cycle impacts of controlled environment agriculture, so that we can do things like reducing the overall environmental impact of controlled environment agriculture. A lot of folks in recent weeks have come out and said that controlled environment agriculture might reduce the distance to the end consumer, but what about all that energy use? If we're using artificial lighting, we really need to try to balance the output of our grow environment and the inputs of our grow environment so that we can reduce the overall environmental impact, minimize energy use. And that's going to take closely monitoring the plant from day to day to understand how the plant is doing. If we can reduce our energy inputs, we can slightly tailor lighting to the life stage of the plant, to the needs of the plant at a given time. Then we can try to minimize our impact. And this becomes a multi-objective optimization problem where we're trying to maximize yield, minimize inputs, and overall improve the life cycle impacts of the grow environment. But in order to go from sensing and understanding the plant to changing the environmental controls with any kind of predictive accuracy, we need models. So the paper talks about the kind of two camps of modeling in the plant field and really in biological modeling in general, which are mechanistic modeling or symbolic modeling, and that's where we have some kind of biological knowledge of the system. We understand through experimentation the basic principles involved in the growth phenomena of what happens when we change certain things. So in plant growth models for the last 50 or so years, one of the biggest, most popular and most well understood mechanistic model that's applied is something called Michaelis-Menten kinetics. And I won't get into too much detail, but the basic concept is if we change the concentration of the nutrients that we're applying to the plant, we have an outcome where we change the uptake rate of nutrients. So this is a model where we understand the physiological phenomenon. We can make a predictive estimation or approximation for what will happen when we change the inputs. And this is a deductive model, an example of deductive modeling. And over the last several years, mechanistic modeling in the plant world has largely been superseded by machine learning models. We have an abundance of data, or at least the promise of an abundance of data in the plant world. We can talk about that later. It's a little sparser than in things like consumer behavior and things like that. But the basic gist of machine learning as it's been applied to plants is that we can use large-scale phenotyping data and big data mining operations, historic data sets from rapid phenotyping experiments and sensor data collection. And we can make a machine learning model where we put the inputs that we're controlling, we put this data into a machine learning model, and we can ascertain how the inputs relate to outputs based on having both input and output data over time, these big data sets. And it turns out that there are some major shortcomings for each of these modeling approaches, and the paper proposes that similarly to how in other areas of biological sciences, we can hybridize these modeling approaches to borrow benefits from each of them. And the benefit of using hybrid models is that, for example, when we are lacking data to produce a machine learning model, if we don't have enough data sets to produce a robust machine learning model, we can use some of that a priori biological knowledge that helped us to generate mechanistic models to fill in some of the gaps. And similarly, we can use data, or we can use big data sets from experimental research to feed into our mechanistic models to better tune our parameter values. So for example, how this one input relates to this output, we can tune those things based on machine learning models. And this is all facilitated, or it slowly is becoming facilitated by biofeedback, the use of sensor data for rapid phenotyping, things like machine vision, and some of the really fascinating stuff Tony will talk about in a few minutes, the opportunity to leverage sensing to produce these hybrid models is really fascinating. And I'm excited to see where the future goes with this space, and I hope to, I'm trying to talk to as many people as I can about it, because we really need data to make models, and that's the moral of the story, is that data sharing is really important. And I'm hopeful that in the coming weeks and years, it will become more prolific, and we'll be able to integrate efforts from academia and the and industry, and come to some more robust models so that we can actually more tightly control the grow environment and more tightly improve the environmental impacts of controlled environment agriculture. So with that, I wanted to thank some of our sponsors for the research. We've been sponsored by the USDA, NSF, and thank you so much to UK UAT for having me today.
Thank you, Abby. That was really interesting. I think it's very important introduction to this concept of DCEA. And I have also sent your paper with a link in the chat for whoever wants to have a quick read, scan read, see what it's all about. So next we have Tony. Tony, Professor of Computer Science at the University of Nottingham, and a few more things and hats Tony is doing. So Tony, would be great if you can share your presentation and talk about phenotyping and all that you are doing.
Okay, so thanks for the invitation to be here. So as Cathy said, I'm Tony Pritmore. I'm a professor of computer science at the University of Nottingham, so I don't know a great deal about plants. I have one from the unkillable range next to me. My background is much more to do with sensing technology, in particular computer vision technology. So I've worked on a number of areas over the years from a computer vision point of view. For the past 15 years, the bulk of our work has been aimed at plant phenotyping. We got started on a project that was aimed at understanding the model plant Arabidopsis, so very much with a scientific audience, if you like, yeah? And it's grown from there. So at Nottingham, we've developed a number of different systems, and there are a number of pictures of them on this slide. Our themes at the moment are deep machine learning, largely for feature detection and segmentation. So in the top right of this slide, you can see a 3D model of a root that was built by taking a root grown in soil and putting it into an X-ray machine in the Hounsfield facility. Next to that picture is an image of the Hounsfield with our technician Brian and our mobile robot side by side. The Hounsfield is a facility that allows us to grow plants in those little orange-brown soil columns that you can see behind Brian and the robot. And the robot can move them around and drop them into a very large scale X-ray machine, so we can X-ray the plant and soil together, and then we have to segment it to figure out where the root is. And you can see the model that we get out of one of the examples on the right hand side. So a segmentation task with the aim of producing 3D models of roots grown in soil using X-ray. We use quite a lot of unusual imaging devices at Nottingham. So the image underneath that is a laser ablation tomography image of a plant root. This is a device which effectively pushes the sample into a spinning laser beam which slices it apart, and with the camera watching the end that's being sliced away. As it's ablated, you get the nice blue image, which if you segment and classify the cells, you can get different types of cell in there. Yeah. We also do stuff with normal color images, and you can see the wheat ear detector to the left of the laser ablation tomography image. So we do a lot of deep machine learning. We try, because we're in the computer science department, to address problems that need some development. So we, by and large, try to avoid just applying existing machine learning architectures. We like things that challenge us. Yeah? So particularly in this area, we get a lot of problems of the same kind of type, where we're looking for something very small in a very large image, and it's hard to get the context that you need to find the thing without having the whole image in. So we've got a number of novel architectures we've developed. We've also done quite a lot on 3D reconstruction from multiple views, and developed a couple of systems for doing that, and more recently have got into active phenotyping. So one of the things that we found with our reconstruction work was where you put the cameras has a huge effect. Yeah? In lots of phenotyping systems, you see people have their cameras on their plants on a conveyor belt which roll past the camera, and the camera position is fixed, whether they're looking at a 6 centimeter Arabidopsis plant or a 6 foot miscanthus plant, which is just crazy because the image quality varies dramatically. And with the complexity of a plant, quite often you need to think about where you put the camera to get the information you need from that specific plant. So we've done a number of things recently, particularly around our 3D reconstruction and detecting features, where we have moved the camera in response to an initial model of the plant to get the views that we want. Yeah, the principle is try to gather the information you need from each plant, the same information from each plant, rather than just take images from the same viewpoint, which is not the same thing. The other thing that I've got involved in over the last 5 years or so is infrastructure and networks. So as Katia says, I'm currently the director of Phenom UK. This is a UK National Plant Phenotyping Network. It's funded from UK Research and Innovation under a scheme known as Technology Touching Life, and the goals of the network are here. The basic idea is that they want us to bring people from engineering and physical sciences into contact with people from the life sciences to try to develop technological solutions that are needed to support discovery science. So Phenom UK focuses on plant phenotyping. Our goal is to make sure that UK scientists have the technology they need by bringing these two communities together, but also to try and understand what the UK needs in terms of phenotyping tools and services. So for the past 5 to 10 years, there has been a development across Europe known as Emphasis. And the goal of this, it's an S3 project, which is the largest form of infrastructure funding in Europe. S3 funds things like big telescopes and CERN, things that no single country would want to buy by themselves or could justify paying for by themselves. And we managed a few years ago to get phenotyping onto the S3 roadmap. So what Emphasis tries to do is to build a distributed research infrastructure for plant phenotyping spread across Europe, so that someone in France who may not want to spend £4 million on the Hounsfield facility can come to our Hounsfield facility and do their experiments there, and we can go and use their field stuff in France. Okay? And the idea is to produce a distributed but tightly integrated research infrastructure. So that's what Phenom UK is about. At the moment we have, and entering our 4th year, 800 members, 850 Twitter followers, the numbers are here. We've done quite a lot of things. We have run a number of annual meetings, obviously disrupted by COVID, but pretty well attended with sort of 50 to 110, 20 people at each one. And we've run a bunch of workshops based around a variety of different things, and you can see the titles here. This is a network that grew out of phenotyping for traditional agriculture. So what we're interested in, or what most of our members are interested in, I should say, not us, is trying to develop phenotyping technology that can be used by scientists or by plant breeders to figure out ways to make better plants. So it's a slightly different environment, and the urban agriculture thing is quite new to me, but very interesting. And I think what we're doing there is reflected in the workshops we've had. Something that is unusual about Phenome UK is a large proportion of our money was given over to funding pilot projects. So we had 270 to 300,000 pounds that we could spend on projects, and we funded 11 over 3 rounds. They covered a variety of things. The disciplines listed on this slide are the ones that our investigators labeled themselves with. Okay, so in terms of bringing people together from different communities, I think we worked quite well, and we funded 13 different institutions around the country. The deal with these was they all had to be phenotyping things that involved some new technology or methodology. So we couldn't fund doing an experiment to measure what happens to wheat when you pour orange juice on it. Okay, it had to be a specific technology component in it. So the pictures down the side are pulled from, I think I'm allowed to say, because they're not here, some of my favorite projects that we funded. The one at the top is we had a group using optical tomography from engineering to look at the textures on leaves, very, very high resolution. The one in the middle is a project from Bristol. What they were trying to do here was use hyperspectral cameras to assess the health of a plant. The problem with that is that if the object you're pointing your hyperspectral at is curved, you get a different response. So ideally, you want to know the curvature of the object before you take your image. And so they integrated a hyperspectral with a 3D reconstruction mechanism so that they could do that. And the bottom one is a field scale thing. And we are working ultimately, I suppose, for field agriculture at the moment, although that's not a limitation put on us by the UKRI. And our remit is phenotyping for everybody. And we just happen to have started from that kind of community. So what this project was doing was putting fiber optic cables under a field, and they developed quite a nice way of measuring the soil water content by sending signals down and looking what's going on. So that's who we are. In the final year of the project, we're focusing very much on emphasis, looking at distributed research infrastructures, and we're always trying to reach out to new communities. And so I think today should be interesting. Okay, thank you.
Thank you, Tony, very much for that. So if anyone has any questions for Abby and Tony, feel free to follow the link Johnny shared in the chat, and you can type your questions, or of course you can unmute your microphone and ask. But I do have a few questions for you guys. So I have actually read that paper of Abby's, and I was thinking about the DCEA, what is all that about? And I was thinking, is there like real-time sensing at the moment available so we could get this dynamically controlled environment agriculture actively, on the table? Or is it something that we are really in a more conceptual level, and a couple of years, maybe 10 years, is going to be doable? Is there such a sensor type? Abby or Tony, it's up to you guys.
The one thing that we can do right now, but it's very expensive, is for things like in my work where we're trying to minimize inputs and environmental impact, is that we can sense things like the environment in real time. We can use sensors in the substrate or the water to see what kind of nutrient concentrations we have, although this is something that's still very, very expensive to do, to look up the individual or to sense and monitor individual nutrient concentrations. We can look at things like the substrate to see the overall salt concentrations in the grow environment, to see how fast things are being taken up by the plant. But this is largely kind of a call to action for sensor development and for plant studies in general, to do studies where we monitor changes in the environment and how the resulting plant phenotype, or those cues, the biosensing, biofeedback cues, can be sensed and then collated with those changes in the environmental control. So it's largely a future forward concept. And I will welcome conversation from the audience as well, if there are experts out there that know more than me in this realm. So that's kind of my point of view.
So I think I have kind of two answers to that, really. One is, yes, you can. There are some things you clearly, as Abby says, you can sense. There are real-time sensors for temperature and various chemical composition things that are out there. In terms of measuring the plant, it depends what you want to measure. There are quite a lot of computer vision based things now that do operate in near real time.
So that's what you get where I'm coming from. So that will, I think. I cannot say
Hello? Right. So there are things out there now that will work in near real time. I think there's a question of what real time means when you're talking about a plant, right? In lots of areas of computer vision, it means 25 frames a second because you're looking at video, and I don't think we're quite in that. But the thing that the phenotyping community to date has done less of is the monitoring, comparing status at one time with status at another time. Yeah, and I think that's an area for development. There's a lot of things in computer vision that do that kind of thing, that track the movement of things, the deformation of objects and so on, but they've perhaps been underexploited. Yeah? So some things we can do, there's a lot more we could do, I think.
Absolutely. And in terms of sensing the feedback from the plant, we can, as Tony is saying, we have a lot of tools at our fingertips to get information about the status of the plant, at least from these visual cues and things like that. But in terms of using that information to understand how changes in the environment will fix those things, like if we see yellowing leaves or things like that, we have a lot less knowledge about what tiny changes in the plant's status mean about the plant's overall health or growth. I think that's the area that we can use these tools and experiments where we change those environmental parameters and then sense what the resulting posture of the plant is. As Johnny said yesterday, what a dipping plant or curling leaves and discoloration, what those kinds of cues mean about the plant's status and what we can do to correct. I think that's where the real research promise is in the future.
Good, thank you. We have actually a few questions in the questions and answers link. And there are two questions about biofeedback. So Julian says and asks, in my opinion, the biggest barrier to biofeedback is the lack of flexibility of growers for real-time control of the environments. Any thoughts? This is what Julian says and asks.
So this is something that we come up against, and I've worked in industry and in academia. I think that this is really where the partnerships, collaborations with academia, between academia and industry is really important. Growers, once they're in business, they don't have the flexibility, as you say, to change things with the hopes that, or with the intention of determining what will happen. We get a good recipe, we get a good lighting condition, and we want to maximize our yield in order to grow and to keep our business alive. The same is not necessarily true in academia. We have the flexibility, we have the ability to tweak things in the environment and see what happens just for science's sake. And I think that that's what is going to be really important, is creating feedback loops between academia and industry in order to facilitate that kind of inquiry and discovery, because as you say, industry does not have the ability to tweak things. Now, the caveat I will add there is there are times when things go wrong in industry. And if you have inline sensors to determine, like, our power went out and this is what happened with the lighting, or if we're sensing the nutrient concentrations and we have a clog or something in the plumbing, in the piping rather, and this is how our concentration changed, and this was the resulting change in the plant status. If we have that information from industry, that can be very helpful to be then unpacked by academic modeling expertise.
Thank you, Abby. And it was also a question of mine, like, which are the challenges to bring together, you know, industry and academia, which you have a kind of answer there. Tony, I don't know if you have anything to add there, but we have a raised hand on the audience as well. Hale, would you like to ask your question?
Yes, sure. First of all, thank you very much for the interesting talk, and thank you, Katia, for getting this organized. My question is really related to what's being discussed, because intelligent learning and logarithmic relationships in control environment systems seem to be very hot topic at the moment, and it's, I think, the way forward. So there's two parts of the story here, the part related to the control environment, to the system itself, and there's a lot of system out there to control individual parameter and to get real-time data. But I think, you know, one of the challenges is to get the sensing technology that provide detailed information about the physiological status, the nutritional value of the plants, the chemical profile, and connecting them with like various computerized or logarithmic relationship. So my question is, you know, I don't have great details about this, but my question is, what's the current status in terms of sensing technology? What sort of tool out there that would provide some sort of details about the nutritional value, apart from the morphology, I mean, the chemical profile and the nutritional value? Are you aware of any technology, the sensing technology there that could provide this sort of information?
So I know that there are all the stuff that I know of, because my background is computer vision, is kind of indirect, if you like, right? I know that you can take little samples of the plant and do the chemical analysis, obviously. The stuff that I've seen that tries to do that is hampered by lack of data to work with. But there are quite a few projects that are trying to do things like relate hyperspectral signatures to the chemical properties of plants, and particularly with things like fluorescence imaging. I know that they have got close to doing that in some cases. I can't really talk about specific ones because it requires more knowledge of plant science than I have. Okay, but the thing that makes that the most difficult to do is that you are looking for a secondary cue. So if you have the internal status of the plant and that affects its chemical composition in some way, and then you're measuring the side effects of that to pick it up, what you need is the data that allows you to build the model. It's a different sort of model from the one Katia was perhaps talking about, but it's a model that links the status of the plant to the image data and measurements you're getting. And I think we're a bit short of that sort of stuff, certainly in my community. Yeah. If, as Abby was saying, you could convince a grower or fund a grower to grow some plants in reasonable numbers and ill-treat them so that we knew what the physical status was and generate the data sets, I think we could go a long way.
Yeah.
Well, just to follow up on this, if I may, Katia, sorry. I work at the University of Plymouth, and I also work in the industry with some private company, and there is great interest in this. So we've got the control environment facility, we've got the chemistry kit to analyze and validate any technology. So if anyone who would be interested to test any technology or to validate data, we would love to talk and see how we can work together on that.
Thank you.
Thank you.
Thank you very much. For my personal interest research I do with strawberries, there is like some technologies, but I think they're in very early age and like times now. They use like spectroscopy, and they can analyze some basically, you know, like protein, carbs and things like that, but they cannot go down to trace elements. And I think there are a few companies that do that, but they need a kind of to get some research, some development, research and development with the specific crop or the specific way of growing and then different cultivars. So I can talk to you later, maybe Highland, we can talk about that if you want.
Yes, sure. Thank you very much.
We have another hand. Hi, David. You can talk if you want and ask your question.
Hi, everyone. Yes, it wasn't quite a question that I had, but I was just listening in. I'm part of UK UAT, and I was listening in just out of interest. I'm part of a company called Altered Carbon, and we're based in Bristol in the UK. And we are a startup that has, we're developing at the moment a sensor technology, which is able to, we're developing a multi-array complex chemical sensor technology, which picks up mixtures of VOCs and gases. And we think that there's some very strong possibility that these sensors can detect plant health factors. So we're at quite an early stage at the moment. We recently received like seed funding for our technology. And we've done some validation projects which have proved our technology in certain areas. So one of those areas was firstly, it was proving the sensitivity of our technology. And this was in relation to a gas called NO2. We also did another project, which was with the National Biofilms Innovation Centre, and we trained our sensor to against a mass spectrometer to detect 3 common biological infections. Now, these infections were related to human health, but obviously we've been looking at one of the areas that we were focusing at the moment in is plant health. And just to give you a bit more background, we actually at the moment are looking for co-development projects to, as you were mentioning on the call just now, you were mentioning there's no sensor technology, and we know that there's not much data out there to work with to train our sensor to become potentially responsive to these very useful, you know, plant health indicators, etc. So yeah, just opening that conversation up really and introducing myself firstly. But what I can do is I can leave some details for you in the chat, and potentially we can set up some conversations with the rest of my team. I'm not from the technology side of things, and from the business development side, but of course, yeah, I mean, you know, it'd be a very interesting conversation to have with the rest of the team and any of you guys. So, yeah.
Thank you, David.
I'll leave some details for everyone. So potentially, if anyone who wants to request our like white paper, brochure, set up any further meetings, then I'm happy to have a talk. Okay,
thank you very much. So we have a few other questions about Tony this time. Tony, a question is, have you tried 3D projection using a fixed camera, but one that can move up and down, left and right?
Projection, you mean 3D reconstruction? Yeah, using a fixed camera that can move up and down, left and right. So we haven't. I mean, in principle, that can work. Hello again, David. That can work. The key thing with reconstruction is that you have different viewpoints. Yeah, so a lot of and it doesn't really matter too much what they are as long as there's some overlap in the images that you get. Yeah? So a lot of the classic computer vision methods for reconstructing 3D from multiple views actually use that kind of, they call it parallel camera geometry, where your camera moves, keeps the axis of the camera parallel and moves like that. So a long time ago, I have, not on plants, with the plant stuff, we had a more flexible arrangement because the problem was much more getting visibility of the features. That we had very complex plants, and to be able to see inside, if you like, you kind of have to put the camera over the top. And so we went for a full 6 degree of freedom robot arm with the camera in the jaws to give us that flexibility. But if the plant architecture is, if you like, simple enough or appropriate to it, you can recover 3D from a simple translational movement. Yeah.
Thank you, Tony. We have one more question from Tao Chen. Hi, Tao. That is for Tony. So, hi, Tony. How do you see the high-end phenotyping facilities can be brought in to in situ analysis to provide the feedback information needed for dynamic CA?
Okay. So I'm not sure that it's a huge leap, really. And I'm not sure I know enough about dynamic CA to be 100% sure, so I may be wrong, but the impression I have is that the real high-end phenotyping facilities tend to move the plants to the camera, right? And why they do that, I think, is because of the size of their facilities and often the size of their plants. If you have a more compact plant, which is what I understand happens a lot in controlled environment agriculture, in a smaller space where you have more control, I don't see why you can't either have multiple cameras or move the cameras around. Yeah? So the core questions for both of them is, how do you get information about the plants out of the images? And I think that's pretty much the same. Yeah. So my feeling is that a lot of the technology will transfer. The thing that's different between the two environments is the motivation, really. So the people that we work for a lot of the time are either scientists or breeders, and they want to understand something on the time scale of a generation of the plant. So they measure things about this plant in front of me now that isn't going to change dramatically in the time I'm looking at it. There isn't much real-time monitoring. Yeah. Whereas in the dynamic CEA stuff, that's what you really want to do. Yeah? So my feeling is there's a lot of stuff that will translate straight away. You probably need to rethink the hardware, but I don't think that's prohibitive. And where it gets interesting is in moving from trying to get information out of a fixed plant at a particular time slot and going to the more dynamic stuff. Yeah? But I really think you can do a lot with that, to be honest. I hope that's an answer.
So we actually have one of my colleagues here, and I don't mean to put you on the spot, Jerry, but Jerry is working on some interesting robotics to do just that, is translate or move what you're talking about. We have these very cool, you move the plants, and the image you shared, move the plant into this chamber where you can take images. And Jerry is I'll let you speak if you're interested, doing some cool stuff with robotics to move the camera and the sensing into the grow chamber. And Jerry, would you like me to put that image up again of the robotic arm?
Oh, sure. Yeah, I also just shared a Dropbox link to a poster that I recently made. I'm not sure if it'll work or not. But yeah, this is still a work in progress for us. But as you were mentioning, there have been a lot of works with like 6 degree of freedom robot arms that can wrap around the plant, so you can take a lot of photos from different viewpoints, Professor Tony. And I guess we want to have a similar thing, but on a robot that can move around to multiple different plants. So our approach at the moment is to use this cable-based design. So this cable-based design can move across an entire wall, maybe reach 100 plants, or if we can scale it up, maybe reach 1,000 plants. And the idea is that we'll have a little robot arm on the end effector of this cable-based robot. So that way we can reach a lot of plants and also be able to reach around and take lots of photos of each of these plants. So we're not, this is still a work in progress, but this is one of the approaches that we're thinking about, at least.
Have you seen the stuff they're trying to do in Wageningen? So there's a big development into a thing called NPEC in Wageningen, where they have the idea of the arm being moved around, but being moved around by a small mobile. So it feels a little obvious in some senses, but they're actually trying to put an arm on a mobile and have it go into different growth rooms and do this kind of stuff.
Yeah, very similar.
I actually think that this is an area that has been for quite a long time understudied in phenotyping, in that people have assumed that you take an image and then the problem is to get the information out of the image. And there have been quite a few examples that you see of people trying to get information out of the image that just isn't in the image. Yeah? So controlling, no matter how you do it, controlling your camera, I think is a much more important than most people think about.
Yeah, that's something that we've been struggling with a lot, which is that if you just have the camera take an open loop path around the plant, then it'll not really capture the correct parts of the plant. You can see, for example, in the photos on the bottom, the plant isn't really centered, and yeah, you're going to be missing a lot of plants that you want to be able to view pictures of. So yeah, we've been struggling a lot with that, and that's what we've been working on for the past 6 months or so.
Yeah, with our reconstruction stuff, the first thing we did was we took a bunch of images. And then we did a really simple segment out the green stuff from the not green stuff, and move the camera so that the green stuff was in the middle of the image and completely surrounded by non-green stuff, and performance jumped. Yeah, a little bit goes a long way in this area.
Oh, great! We're on the right track. Wonderful.
Can I just echo to the the discussion that has been led by Tony and Jerry, because maybe the other angle is the practical needs, whether we need that detailed information to be provided for this controlled environment. Just to make an analogy, because as a process control engineer, in the past we worked for process control of pharmaceutical processes, and historically to analyze pharmaceuticals, you use this very high-end equipment and sensors, which can only be done in a central lab. And gradually, because of the economic needs, people move those sensors into the in situ analysis. So you can actually get information on the spot, and that provides the feedback information. And then you can use that information in your mechanistic model, in your machine learning model, to devise your control algorithm so that you can dynamically control the environment parameters to achieve the best you can hope for your process. And it seems to me we're seeing a similar trend in the controlled environment agriculture. But I guess how fast we can go along the way. And whenever you introduce new sensors, it's additional cost to the growers. So how fast we can go along the way really depends on the economic needs, I think, ultimately. Yeah. But as academia, as people working in academia, we can always look forward to see what is going to happen in 5 or 10 years' time.
And in order to get funding in academia, it really helps when industry practitioners can say, this is a problem that needs to be solved, because in academia, we can find problems everywhere, right? So knowing that this is a problem, and what you're saying is this really necessary? Is this a necessary thing to be sensing? It helps justify the academic study as well. And one thing that's really going to be interesting is the more information that we get and the better our modeling capabilities become. I think we'll find that we can use a lot fewer data points in order to get a good picture of what's going on. So right now we're talking about, all of this morphology about the leaves and the plant structure and the color and everything, but we might find that it takes a lot fewer different data points to get a good idea of what's going on in the plant once we can build a good, robust picture of what's going on, then we'll see that, oh, well, actually, we didn't need to know what was happening at the tip of the leaf. We just needed to know the color or something like that. And we'll be able to pare down the sensing requirements to target the very specific things that we need to do. But this is a ways off, and it's fun to think about, but to really drill down at, it's going to take a lot of data.
So there are some things like that happening in the broader computer vision community that are starting to get used in plants. So an awful lot of the computer vision stuff at the moment uses convolutional neural nets, which you find and you train it up to do what you want it to do, and then it does it as long as you ask it the right question at the beginning. A lot of people have concerns about this being a black box, so it may well be that we can train a network to tell the difference between you and me really easily, but what's it doing? It may well learning that the difference isn't between a man and a woman or whatever, but it's the fact that I've got a guitar in the background, and you haven't. So there's a growing literature on analyzing these networks to see what it is that they use. And what you can do now is, we've done it with some soil science stuff recently, we've trained a network on X-ray data of soil samples under different conditions, and trained it up to be able to classify them, so it can recognize type A from type B soil. Then gone to the network and started at the output and worked back through the network, looking at which parts of it were active when the decisions were made, right? And the classic, and you can see which features are being used to make the decision. So the classic example of this in broader computer vision is the first people to do it trained a network to tell the difference between cats and dogs. And when they visualized it, they realized that what it was doing was cats have little pointy ears and dogs have big floppy ears. And that's what it was actually basing it on. Okay, so you could put something in with hold up two triangles and it says cat. You can do quite a lot with this now.
Well, I'm excited that you brought this up, because this is really the crux of the hybridization justification. And it's that typically, or when we talk about machine learning, specifically when we talk about deep learning, when we're essentially just feeding a ton of data to a machine and expecting it to get some kind of, to ascertain some kind of correlation between these inputs and outputs, we do treat it a lot of times like a black box. And some folks in the artificial intelligence realm and in the deep learning realm have said, well, let's go back to some of the early foundations where we have symbology being the foundation of learning models, where, and this is where mechanistic models become very helpful. We know stuff about plants. We know that a change in one input is going to likely lead to a change in a certain phenotype or output. And so we can build machine learning models. And this is a debate that's pretty hot right now, I think, is the integration of these symbolic models into the building of machine learning models, because we do know some stuff that we can build our models based on instead of treating it like a black box.
Thank you, Abby. I have a very close to what we are talking about now question from the audience. So could you give some examples on which models worked better with machine learning approach and which with mechanistic approach, and which worked well combined?
So this is a great question. I think that in the plant realm, I haven't found very many, and I'm open to being corrected, but I haven't found many combined models. But in human physiology and in the study of enzymes, there have been a few studies in the last 5 years or so where scientists have combined mechanistic and machine learning models into these hybrid models. In the plant realm, mechanistic models were the prevalent mode of predictive modeling in plant science up until probably the early 2000s. And more recently, a lot of folks have turned towards machine learning models because of the kind of buzz around them and the excitement about these new sensing techniques. But in, I guess I'll speak specifically to the hybrid models in physiology. There have been some studies about protein folding, which is really cool. And since we know something about the shape of proteins and we know how different different active groups or proteins are really big, and there are a lot of different chemicals sticking off of proteins that interact with one another. And since we know a fair amount about protein folding already, and we know something about how these little functional groups that stick off of proteins interact with one another, we can feed the model information about that. And then we can use machine learning to look at a whole lot of different proteins and understand how they're folding. And a lot has been done in this realm in the last 5 years. So I think that it hasn't been yet applied on a large scale to plant modeling, but I'm excited to see where it goes in the near term.
Thank you, Abby.
I know that didn't really answer the question to the full degree, but I'm happy to talk about that at another time. It's just, it could be a lot of.
Yeah, maybe it's a bit complicated. I know that's a kind of conceptual what you are proposing there with DCEA, but yeah, a bit. So I have a few more questions. I think I'm going only to ask one more. That is from Enrique. A bit different question. I don't know if we know the answer. So it is true that plants communicate to each other by some way of chemical signaling. And if it is, could you use a specific sensor to track it?
So I know partly the answer to that, but it is only partly, and the answer is yes, they do. I have seen examples, or I've been told by my plant science colleagues of examples where things happen like a predator, an insect comes over a field, over the boundaries of a field and attacks the leaves of one plant. That plant exudes something through its roots, which are picked up by the neighboring plants and taken up into the shoots, which then give off some kind of mild insect repellent. Okay, that is my total knowledge of it. I know that it does happen. Yeah, but whether you can sense it, I really don't know.
I think the key here is that you need to know what you're looking for. There's a lot of noise that might confuse sensors. If you don't know what chemical indicators that you're looking for, it's very hard to sense them or to even build sensors to detect them. And I think that that's a limitation right now. There's an interesting book, The Secret Life of Trees, and that talks a lot about signaling between trees in the forest through the rhizosphere, the root structures. And I think it's a fascinating realm that will, as we learn more about what happens between plants, we'll be able to develop sensors to detect these signals. But until we know what the chemicals are, it's very hard to know what to look for.
Brilliant, thank you. I think we have reached to the end of this panel. If you would like to get in touch with the panelists, please feel free to either talk to them directly, or if you want their details, you can get in touch with myself through UK UAT, Mark or Johnny, or whoever you want through UK UAT. It was quite interesting. I'm really looking forward to see how DCEA is going to evolve in the future. And Tony, how phenotyping can help with CA and urban agriculture. We know that lots of people in our membership, they are currently using phenotyping technology in their vertical farms. So I'm sure that's going to be more interest about what you are doing, Tony, in the near future. So thank you very much again. You can find this recording shortly, potentially within 5 days, in a week, on our YouTube channel. And thank you very much for everything, everyone. Thank you, Tony. Thank you, Abby. Thank you, Johnny. Bye-bye.
Thanks so much for having me.
Bye.